Good afternoon, all friends. We are from Group 5. We will present the result of our work entitled Plant Disease Epidemiology. Next, our group member are number one is Ummun Zakiyyah, student number 105, and then Veronika Estika, student number 108, Fauzan Ridlo NA, student number 110, Yuli Utami, student number 170, Zalfa Nahda, student number 137, Marlena Hilda Sari, student number 143, and Taufik Kurniawan, student number 144. Next.
Definition. Epidemiology is the science of disease in population. Epidemiology that is often used in the change in the intensity of plant disease attack in a plant population at a certain time and space. Next. That is important to note from this definition. It is emphasis on population phenomena. The important population involved in an epidemic are the host population and the pathogen population. Epidemiology discusses how two populations interact with each other, resulting in a disease that causes yield loss. This interaction is certainly influenced by the environment in which the interaction occurs. This environment includes both biotic and abiotic. Humans can influence the interaction between host pathogen and the environment, and in fact, this influence is increasing day by day. Thus, epidemiology also discuss the influence of humans on plant disease and sometimes the effect of plant disease of human life. Another element of epidemiology that is no less important is time. Time affects other elements. Plant disease can only occur in the presence of sustainable plants, virulent pathogen, and a favorable environment occur at the same time. However, in epidemiology, it is not just a disease caused by endemic, but more broadly, it involves all forms or change in the level of disease attack and the factor that influence it. Next.
Spread of pathogen. The spread of pathogens can occur actively or passively. Passive spread that plays a major role in causing disease, namely by wind, air, animals, especially insects and humans. Some pathogens can actively spread, for example, nematodes, zoospores, and bacteria.
Factors. Number one is wind. The spread of disease by wind is airborne, such as spores of pathogenic fungi on leaves, stalk, and fruit. Number two is water or rain. The spread of disease by air or rain is airborne or waterborne. The presence of water or rain on the bacterial exudate causes the bacterial cells to disperse and spread to various contact points or points of penetration. Number 3 is insects. Insects will deposit or leave the inoculum they carry to the injured plant part or because of the insect's feeding process. Next. 4 is seeds and other plants. Pathogens or inoculums or fungal spores or bacterial cells are often carried in infected seeds or plant buds, so they can spread the disease to other areas or to other plants. And 5 is human. Humans are a long-distance spreader of plant disease through the introduction of seeds or plant but infected with pathogens between countries or regions.
And then is disease cycle. Disease cycle is disease cycle involving change in plant and symptom plant and change in the life of pathogen in length of period in one season or planting, and from one planting season to the next growing season.
Next, disease cycle consists of 1. Inoculation, inoculum contact process with host plants. At this time, successful landing or arriving inoculum to plants parts or condition pathogenic predisposition greatly determines the stage, the beginning of the disease process. 2. Penetration, entry of pathogen into host plant tissues, for example, through cuticle, epidermal cells, or intercellular spaces. This process starts with spore germination or emergence of tubes which later develop into a structure that plays a role to attach themselves to the surface of the host. Then comes a hosterium, which is a structures or organ that play a special role take food. 3 is infection. The process of the pathogen making contact with sensitive plant tissue cells and take food from him so that disease occurs. For infection to occur, the organism must be in a pathogenic state. The host plant is sensitive and the conditions are appropriate. The interval between infection in plants and the one set of symptoms of the disease is called the period incubation. Next. For infection, for fungi will grow mycelium in cells in the cuticle tissue, epidermis, or other tissue, or it can also envelop surface of cells. At this time, mycelium can spread or grow develop intracellularly or intercellularly. Bacteria as a single cell and viruses attacked intracellularly or individually carry out growth in the cell, while fungi can grow from one cell to another cell. 5. Growth and reproduction. Pathogens grow produce normal body structure used for the survival of the species in outside the pathogenicity system or for grow and life in a pathogenicity system that new or an new unknown host as an inoculum such as spores, mycelium, conidia, sclerotium, and chlamydospores. And the last one is dissemination. Sorry, this is wrong. The right is dissemination or spread of inoculum. So transfer of pathogenic inoculum from a source in the affected part of the host pathogens to other hosts. Next.
Plant epidemic forecast. Forecast of disease if the epidemic comes can be predicted with a sufficient period of time to carry out prevention efforts. Next. Most epidemics are mainly determined by weather factors that are difficult to predict, and only a few diseases have known determinant. So only a few diseases can be predicted as a epidemic. In addition to observing weather factors such as air humidity, sunlight, biological observation are often required, such as the density of pathogenic spores in the air, insect vector populations, and others. The more complete the available data regarding the relationship between disease intensity and these various factors, the more precise the forecast will be.
Then, to develop a forecasting method, it is necessary to know which stage of the disease cycle plays an important role in the disease. Two, for monocyclic disease, in predicting developing plant disease, they must understand certain characteristics of the particular pathogen, host, and environment in general. Three, for polycyclic disease, such as late blight in potatoes that have a small initial inoculum but have multiple disease cycle, disease progression can be best predicted by estimating the disease cycle rate. For this disease with a large number of initial inoculum and disease cycle, such as beet yellowing, both which is initial inoculum and disease cycle rate must be estimated for accurate prediction of the epidemic of the disease. Next. Epidemic formula. Epidemic formula, first, simple interest disease or penyakit bunga sederhana is XT or final inoculum equal initial inoculum adding initial inoculum multiple R is infection rate, multiple T is duration of the epidemic. Then penyakit berbunga majemuk or compound interest disease, the formula is XT or final inoculum equal XO or initial inoculum, then multiple E or constanta, then exponent R infection rate, then R multiple T, which is duration of the epidemic.
Uh, okay, I think that's all. And if you have any question, you can write your question onto the forum discussion column at Spada, and we will answer your question as soon as possible. And thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.